So hi everyone, and thank you, Christophe, and I want to thank, of course, all the members of the jury. So thank you, Takashi Toyofuku, thank you, Emmanuel Gélin, Cyril Marchand, and Nasa. You've all been introduced. Thank you, Christophe, for accepting to be at the head of the jury today. So yes, I'm going to present this habilitation, which is going to, of course, talk about the work I've been doing the last few years on geochemical signatures of marine biogenic carbonates. And so in my case, it's exclusively foraminifera and corals, and as paleoclimatic and paleoenvironmental proxy. So here we go. This is a brief summary of the defense I'm going to give now. So first I'll start with my career summary. To show, what was really the path that got me where I am today. Then I will briefly introduce the general scientific positioning, then the early contribution and multi-proxy development. So this is mostly my PhD work. And then I will talk about expansion into new proxy, and that is literally the work I've been doing during the 2 postdocs I've been doing at the end of my PhD before I was hired at the ERD, and how much this all, kind of led to the position I'm having now. And then I will present a selected contribution, and I will finish with perspectives, and mostly presenting 2 projects that just got funded this year and that are going to be the main research activity over the next few years. So this is it. So to start with my formation, so I had my high school level, my baccalaureate in French, which was in I started university in 1999, and I started university in my hometown just after that. So it was Clermont-Ferrand. So this is the picture of the university you can see here, the Université Blaise Pascal in Clermont-Ferrand, where I spent 3 years, where I did the equivalent today of the bachelor. And I did a bachelor in biology and geology. And after these 3 years, and I really enjoyed that, but I realized I really wanted to work in marine sciences. So I decided to move to Bordeaux in the southwest of France, where I started a master called ENVOL, which is literally a master in oceanography. So I spent 2 years in Bordeaux doing that, and I did my 2 research practical every year at the end of your master. You need to do a research practical, and I had the chance to do both of mine at the University of Angers under the supervision of Professor Franz Jorissen, and it's where I started working with foraminifera, because he was a very big specialist of foraminifera, and he still is. And he introduced me to that during these 2 research practical, and I did that the first time, and I decided to keep doing that the second year because it was really fascinating. So it all started for me at that point, really. And between year 1 and year 2 of my master, I actually had the chance to do an internship at the NIOS, which is the Netherlands Institute for Sea Research. Sea Research is the Royal Netherlands Institute for Sea Research, which is basically the largest research institute, marine research institute in the Netherlands. And that was very, for me, it was a key point, because after that, I decided, okay, this is really what I want to do. I want to move abroad. I want to keep doing this type of research, but overseas. And so it's exactly what I did at the end of my master. I moved to Germany, where I started my PhD at the Alfred Wegener Institute. So that's the picture you can see here. So it's a very large research center in Germany, and I kept working on foraminifera, but I changed a bit my topic, as at that point I decided I wanted to change the type of proxy I was working with, and I started to look at geochemical signature, and actually I kept doing that my whole career afterwards. At the end of my PhD, I started straight away a postdoc in Paris. I switched a bit organisms. I went from foraminifera to corals, and then I switched also elements, the type of proxy, but I will present all of that. And I did actually 2 postdocs. They were interconnected. So one was in Paris, and the second one was like 3 years in Perth in Western Australia, and that was the continuity of the first one. And before the end of my second postdoc, actually, I was hired by the ERD. It's a national contest, so I did this contest before the end of my contract, and I could negotiate to finish my contract in Australia before starting my job at the ERD. So this is what you see. At ERD, I'm a part of a big research lab, which is called the L'Océan, which is located in the center of Paris, and mostly in Sorbonne University. This is the Jussieu, for people who know that. And there is a second location for this research center in Paris. It's in north of Paris. It's called Bondy, and it's only ERD. And this is this one, and it's actually being closed right now this month. I think it's now being closed, and all the people are going to move to the very center. But anyway, I've been working there for a couple of years. And so that was it. So I want to give a few words really about my research lab. So I'm part of the L'Océan. The L'Océan, it's a pretty large research laboratory. The title really stands for Laboratoire d'Océanographie et du Climat. It is about 180 people, among which you have 180 people, among which 120 scientists. And the main expertise, I would say, it's about ocean physics, marine biogeochemistry, climate science, ecology, and numerical modeling. So it's all about ocean science. It's pretty diverse. It has four main, what we call tutelle or head laboratory, if you want. The main one is Sorbonne University, which is the host in the center of Paris, but you also have ERD, CNRS, and Museum of Natural History. And interestingly, it belongs to a much broader research federation, which is called the IPSL, the Institut Pierre-Simon Laplace, which is getting quite famous now internationally, which is actually gathering several labs focused together, and they all work on climate and Earth system science. So if you hear about IPSL, my lab is part of that. It's not the only one. It's pretty large, but it's interesting. Anyway, within the L'Océan, you have 6 research teams. I'm not going to explain everything. And among which I was at the head of one, the fourth one here, called Valco. For 5 years, I was co-leading, because we're 2 people at the head of this team for 5 years. It's a pretty large research team, and it's a pretty time-consuming activity, but also very interesting. And just to finish on the lab, he also is leading a few national observation services, and among which the first one, the SNAP-CU2, which is a national service for oceanic parameter analysis, among which the CU2. And for the science I develop, it's super interesting. It's like a lot of data that are readily available, and it's very, very interesting. Okay. Sorry about that. So now I keep going with my career summary. So I will give a short overview of that. So in terms of publication and congresses, so I've published since the start of my career, so the start of my PhD, basically 31 peer-reviewed articles, among which 12 as a supervisor, which leads to an impact factor of 19 for citation of 1,600. Those are the latest Google Scholar things I could find on the internet, I think 2 days ago. Anyway, I participated in many conferences and workshops, about 50 of them, and a lot of national, but also a lot of international ones, among which some large and pretty famous in terms of geochemistry, such as the Goldschmidt, but also the EGU, the ICRS, the ASLO. And for 3 of these congresses, I acted as a chair, twice for the ICRS in particular. Anyway, so I was also part of a few research cruises, and some pretty major one. And the one I listed, that's only there's 2 of them on Icebreaker, the Polarstern and the Aurora Australis. And I listed those ones because this is 3-month cruises we're talking about. So this is pretty major. And a lot of the data and the research I published afterwards were, of course, from the samples I could collect during these long research cruises. Okay, so I keep going in terms of research projects. Since I was hired at ERD, so literally since 2013, I was part of 19 research projects, either as a PI or a co-PI, which is about, so I did a rough calculation, which is about an envelope of €2 million over 12 years. Yeah. Of course, through a lot of collaboration, national and international, and I listed here the one that I still have very active today. So with the NIOS, again, in the Netherlands, the Alfred Wegener Institute in Germany, the University of Sydney, UWA in Perth, Australia, Lamont Doherty in New York, ZMT in Bremen, Germany, the Jamstock in Japan, Nanyang University in Singapore, and a lot more. I mean, this is just like the main one I'm working with, like basically on an everyday basis. And more than 50% of the 31 paper I cited were with international co-authors. I think it's more than that actually, but at least 50%. In terms of scientific leadership, I'm not going to detail everything, and as time goes on, obviously, as a scientist, you are always more involved in committee and in a lot of things. But the main one remains what I cited just before, to be at the head of this research team, the VALCO research team. At the time, there was 17 permanents. It has reduced a bit since then, but was 17 permanents. And also, it was within this research team, you had the people that were leading the analytical platform ALISE, which is a pretty large geochemical analytical platform, and with all the problem that goes with that, because you have a few mass spectrometers and so on. So it was a pretty, it's interesting, but it's a lot of, yeah, I mean, it's interesting. So now, in terms of students I supervised, so since 2013 still, I supervised 12 Master 2 students. One of them is about to start, so I would say literally I supervised 11 plus one starting next month. 4 postdocs, one is in progress, 3 PhDs, one is in progress as well, and I was a member of 2 PhD committees. Over the last few years in particular, I've been the reviewer of a lot of numerous scientific journals and research calls, like as soon as you're hired basically on a permanent position, you get all those projects to review on a regular basis. But over the last few years, I got involved with the European Commission, and it's interesting because when you start, it's actually I realize that they keep coming, and it's again very interesting. But it's yeah. So I've been working twice as a reviewer for the Marie Curie postdoctoral fellowship, and also once for the Slovakian Research Agency, which is the equivalent of the ANR but in Slovakia. And then I also acted as a reviewer for the CNF-ASH, which is the National Offshore Fleet Commission. So it's basically to evaluate a sea campaign request proposal. And in terms of teaching, and I'll finish on that, so I am not a lecturer, so I don't have hundreds of hours, as you can see on different profile. But the last few years, again, I've been involved in the new master that opened here in New Caledonia, which is called the Semester on Sustainable Development of the South Pacific, and I particularly teach on ocean acidification. And that, so I've been doing that at least 3 times. Every time, like, because the master doesn't open every year, but every new time it was opening, so I did that. And also this year, finally, I was part of this summer, like university summer school, if you want, in Vanuatu. And over a week in Vanuatu, which was very interesting, and that's it. So I'll finish on that for the career summary. Of course, I want to say that all of this, whether it's publication, whether it's congresses, whether it's all the work we do, it's the results of many, many collaborations. So I started to try to plot all these pictures of all the people I've been working with at one point or another, and then I kept going. And then I kept going, and then I stopped, and I said, okay, this is not a good idea. It's never ending, and I want to thank all of those people. And more importantly, I want to say that this is probably one of the things I prefer in this job, is that there is always someone new coming, someone with new ideas, someone with a different background, and this is so rich. And this is what makes it very interesting, like they keep being interesting. So I thank all of the people. I am sorry for all of the people that did not appear here, but again, there's like too many, and so I hope it will keep going exactly like that. So now I'm going to talk about the general scientific positioning of my work. Sorry. So for me, again, it all started with foraminifers. And I put it here like a lot of pictures, many different pictures. They are all foraminifers because they can be benthic, they can be planktic, they can be open ocean, deep, like they can be very deep in the ocean, they can be on the coastal. And I put all of these pictures because I was fascinated by them as soon as I started working with foraminifers. And I still don't understand why everyone is not in love with foraminifers as much as I do, but okay, I understand. So this was the start for me of the scientific journey, really. And like I mentioned before, it started during my master, and I kept working. So this is the path, but the whole path I kept working on carbonate geochemistry, and I use proxy research like based on environmental information recorded in marine biogenic calcium carbonate, and again, foraminifera and corals. Anyway, the motivation for this work, so now I'm at the, let's say in 2005, at the beginning of my PhD. So at that point, we're starting to get a lot of data, and we know that we're facing global warming. We know that the data are pretty clear. So this is a very recent graph. It all comes from the IPCC report, but already at that point we have like pretty good data, and we knew we were facing perturbation. We could see already both the ecological and climatic impact with, so I put like an example of coral bleaching, the coral that started to be bleaching a lot, and we had people facing both inundation and drought, and so that was the first perturbation, and that's where a lot of science was being funded to try to find not necessarily solution, but also better understanding and monitoring of what was happening. At the same time, I was going to say, and in the following years, we also started working, and we heard more and more about ocean acidification, which is the evil twin of global warming, and just a direct consequence of the CO2 going back from the atmosphere to the ocean, acidifying at the same time, first the first layers of the ocean, and then everywhere. And so that was exactly where actually the subject of my PhD started, and where the funding came from, with this double perturbation of both global warming and ocean acidification. Of course, now that the time keeps going, we are facing a triple perturbation with acidification. Now we can clearly face and observe ocean deoxygenation. So, and I stopped here, but there'll be more. I'm not surprised in 5 years, in 10 years, there'll be something new, another study about something. But anyway, already with all of that and all we could observe, that's where the proposal and the funding were coming from when I started my PhD. So with this planet being a bit mistreated, I would say. So now the question was, what are our approaches, sorry. So the idea was very quickly and very often to look in the past, to look in the idea why paleo, because with paleo, what has happened once can happen again, so that's the basis, obviously. But it also, it provides key information, that's what we are seeking. But also the interesting part with paleo is that you can basically pick the type of climatic anomaly you're interested in. So if you want something with a very, like, let's say, something happening very strongly and very quickly, you'll have this type of anomaly. You can have a climatic anomaly happening very strongly, but on a much longer time period, or on a long time period, but a lot softer at the same time. And that's a bit so, like, for example, the first one I put here, like this is the Cretaceous tertiary boundary carbonate crash. This is the disappearance of the dinosaurs. So here we are 65 million years ago. And what had impacted, obviously, like dinosaurs, also had massive impact on a lot of things happening into the ocean, and among which the foraminifera. And this is what you can see in the sediment. We had this carbonate crash, and it has this name for a reason, because you had this drop in carbonate arriving at the bottom of the ocean that was absolutely massive. This is here. And not only you had a flux that was very reduced, you also had the size of the particles that dropped like, so you see, this is what was, do you see my error? So this is what was arriving, the size of what was arriving in the ocean before the crash, and then what was arriving afterwards. So that's the type of anomaly that is very interesting when you work with sediment core, obviously. And then you have this one. Then the question was, what can we find in the past that is mimicking the best what we are experiencing today in terms of so temperature raising, ocean acidifying, and ocean loss of oxygenation. And actually, the best one we can find in the history of the Earth is the PETM, the Paleocene-Eocene thermal maximum. This is what you're seeing here. So here we are big. We are not at the same time. We are about 56 million years ago. And the PTM is particularly interesting because you are facing a massive cooling on the planet everywhere, and then suddenly you have these hyperthermals, meaning everything is cooling, and then suddenly you have this rise in temperature, this loss of, this rise in temperature, this rise in acidity, and this loss of oxygen happening all of them at the same time, exactly what we are experiencing over, of course, not the same time scale, over 20,000 years when we are about 200 years, but still with the same kind of changes that we are seeing now. So all of that are the reason why we are looking into the past, why we are looking at the paleo, because it gives us keys and information that we cannot gather otherwise. So now there was the question, the why, and now the new question is how. So one of the tools we have is the geochemical proxy applied, so in this case on marine biogenic carbonate. And I'm citing that because for a very long time, actually, the magnesium calcium of foraminifera, that was the one and only information we had about temperature on very long time scale. So this is before the magnesium calcium, you had the oxygen isotopes, and this is still a tool that is widely used today. But again, we're talking about timescales where we don't have a lot of choices. What do we have for information? How can we get all of this information? So magnesium calcium for foraminifera, it was a very major one. But as the time is going and as the technology are evolving as well, we have today like a toolbox which is growing, which is always growing with new proxy happening every day. And this is really something I've been working on a lot. So the idea is, again, to reconstruct past climate and past environmental condition. We use this elemental concentration and isotopic composition on these marine carbonates. So how does it work, basically? So you have the input. The input, in our case, that's the environment. So that's the temperature, that's the salinity, that's the concentration in heavy metals. That's what you want to reconstruct much after the moment that this has passed. And then you have the output. Again, in our case, the output is the carbonate. So that's the foraminifera or the corals, whichever you want. But the problem is the put. And the put, the little chicken here, it's this black box, because we tend to say, okay, for a long time, people have considered, oh, it's just a stone, it doesn't have, it's okay, it's just passive, it doesn't have an impact. And we realized very quickly, actually, about 40 years ago, that no, the put was the key, because, because we really need to understand what's going on with this black box if we really want to use this tool. And this is exactly what I did during my PhD, so early contribution and multi-proxy development. So that was exactly that. So I started my PhD with the first principles, which is that foraminifera shells, which precipitates in warm waters, they tend to contain higher level of magnesium compared to the counterpart that have precipitated in cold water. And this was happening at the same time that the oxygen isotope proxy was being developed. So we are in the 1950s or something like that. And while the oxygen isotope was giving a lot of very good data, the magnesium calcium in forams, it wasn't doing very well. And we could say, okay, well, we have that, but it's not functioning very well. Until this paper in 1996, where suddenly what they did, actually, they changed the way how to treat the samples. Instead of taking like to grab some sediment from a very clearly dated core, they said, okay, we're only gonna work on one species on a very on a short size fraction, because we can see that there is a lot of different impact that we did not consider before, which is like ontogenetic cycles. This is going to have an impact on the geochemical signature, and in that case on the magnesium calcium concentration, but also not whatever species. Some species have symbionts, some do not. We cannot mix them up. So anyways, that was the start of the idea to develop a mechanistic understanding of elements incorporation to improve this proxy. So that's exactly what my PhD was about. And for that, you literally have two possibilities. The first one, it's through culture studies. Why? Because when you do culture experiments, you literally have control on everything. You know every single parameter. You can measure everything at the start, during, and at the end of your experiment. So this is ideal. The second possibility, and what you need to do always anyway, that's the field verification. But, and that's a big but, on an environment that's extremely well monitored. Again, you need to get as many information as you can if you want to improve this mechanistic understanding. So that's what I did during my PhD. I worked mostly on this species, Ammonia tepida, because it's a good, how to put it, it's a good lab rat for foraminifera. That means it's a species which is going to survive very large variation in salinity, in temperature, in everything. And also because they do not die in culture. I mean, not straight away, which is quite a big parameter because the others tend to die very quickly. It's pretty hard to keep them alive in culture. So I work with this species. I'm not going to detail everything. I work with this species during my PhD. And one of the first things we did was to be able to try to recognize what part of carbonate is actually precipitated into the culture experiment. And for that, we use a dye. This is what you see here, like this yellow fluorescent dye. And when the foraminifera, they produce carbonate while they're bathing into this dye, in the condition that you're interested in, they produce carbonate which is stained like that under UV light. You can easily recognize a chamber that has been produced in your experiment. This is the bright green thing. And why does it matter to us? Because at that point, we were already capable to analyze the geochemical signal of one single chamber of foraminifera. And this is what you have here. Because we had the mass spectrometer, they had developed enough that they could be coupled with laser ablation. And laser ablation, it allows you to target literally surfaces of carbonate that are 40 microns in terms of diameter, which is nothing. And in terms of paleoclimatology, when you're trying to reconstruct climate and you're using foraminifera from cores covering millions of years, I can tell you that being able to determine this type of geochemical signature on one single shell is a huge thing, because often you have three. So if you have three, that means you have 3 points and you start having data where otherwise you literally have nothing. So that was the first paper of my PhD. The first paper was to check actually that the dye was not going to impact the geochemical signature. And this is what I did. And then I did different type of culture experiment. I checked on the impact of different parameters other than temperature on this proxy. So first I looked at the impact of the salinity, and then I looked at the impact of pH, all of trying to deconvolve what these other environmental parameters have for an effect on the geochemical signature. So, and then there was another paper with field experiment. This is what I was telling before, because that's a bit the only two things you can do. But anyway, this is what went out of my PhD. And so now I'm going to start to move after the end of my PhD and expansion into new proxy. So I finished in 2009, and I decided quickly at the end of my PhD to use this kind of pretty dense carbonate chemistry background I had to learn to do all this work and to try to move on to something a bit different. So I went into a new proxy, and not this time I was looking into pH reconstruction, because again at that point, I don't know if you remember, now we were talking about ocean acidification. The 2009, it became very fashionable, ocean acidification, and with fashionable topic comes, without surprise, findings. And so that's what happened to me at the end of my PhD. So I moved into that, and I did these two postdocs, the first one in Paris and the second in Australia. And not only I changed proxy, I also changed bioindicator or archives, if you want. I went from foraminifera to corals. For the first reason was because the quantity of carbonate you could get from a coral core is a lot more important than what you get as a foraminifera, which is extremely small. And for proxy like boron, which is an ultralight element, it's actually a good, technically speaking, it's very advantageous to have more material to start with. So anyway, I moved into corals, and I discovered a whole new world in terms of how they calcify and they produce their aragonite. And at that point, I also had the chance to be part of a paper because this is really, these few years of postdoc, I really spend my time really working on new proxy development and also on proxy calibration. And I had the chance to be part of a paper who developed this new proxy called lithium magnesium, allowing for temperature reconstruction on coral archives. And that was a bit of a growl, this proxy, because it appeared to be very insensitive to the vital effect, meaning the bio impact of the coral itself didn't seem to impact this proxy, which was a big surprise. So it's exactly what you can see on this graph. You can see that you have the same correlation between the concentration on lithium and magnesium and the temperature, whether you're working with cold water corals, temperate corals, and tropical corals. Of course, they are very different organisms, and still they were all responding with the same, following the same correlation with temperature. So that was the start of my postdoc. And then so at the same time, I work also with boron isotopes, and that's an example of, actually that's probably the only data you'll have in this whole presentation that have not been published. But this is data I got from a core from Clipperton, which is a pretty crazy island in the very east part of the Pacific. And what we get from that, we got this coral core, and what you could see here. So this is like 80 years ago, and this is today, and you could see a clear acidification. Oh, sorry. You could see a clear acidification over the last 80 years, and at the same time, you could see a clear increase in temperature using the lithium-magnesium ratio. So that was what I started doing during my postdoc. Of course, all of that, and I mentioned that before, was also because technology speaking, now we were capable to get there and to do that. And little by little, and that moment exactly, the boron isotope technique we could apply to corals, we were capable slowly to apply them to foraminifera, again, thanks to laser ablation. So this time, coupling laser ablation with multi-collector ICP-MS. So here we're talking about machines that are something like €2 million each. And so it's usually, of course, it makes sense to say that those projects, it's only feasible because you have a lot of different labs that are involved. When you have such expensive machines that are so complicated to maintain and to run, you can only work with many, many different labs together. And this was already the case then, and it's still the case today, and that's why I started to list a few labs. Anyway, so I kept going with my postdocs, and so I worked on many different things. I looked, for example, at the impact of dissolution on the geochemical signature. So that was the first paper where we looked at the impact on dissolution on the boron calcium signature. And then I also looked at the impact of light on coral boron isotopes, again, through culture experiments. So there was also it's pretty challenging because it was a completely different world, but it was very interesting to try that. Then I looked at the impact of the type of mineral, for example, on the geochemical signature. So this is an example. This is a coral head. This is a slice of a coral head. And it's very interesting because this coral head, very briefly, was growing very normally until it met a cyclone, the Ivy cyclone, pretty famous, all from New Caledonia. And it probably rolled over on the side, and it kept growing after a while. And instead of precipitating aragonite, it started to precipitate calcite, or at least mostly calcite, when before it was just aragonite. And we looked at that. We looked at this impact of change of mineral on the geochemical signature. And it was drastic. It was not a surprise, but it was very interesting to see that. And then I also looked at something more basics, like the impact of the type of cleaning and treatment we apply to our samples before we analyze them. And all of that has an impact. So all I want to say here, that this is the kind of work you do here. It's infinite, and it's extremely interesting. But it's also like you really need to work on a very, very precise way. And that brings me back to this very first slide I show you with the foraminifera. We cannot treat our samples. If we want to have data that are actually, which makes sense, we need to treat our samples in a very, very precise and defined way, whichever element parameter you're trying to reconstruct. Anyway. So the work I presented before led for me to be hired at the ERD. And since I was hired at the ERD, so since 2013, I kept doing, among other things, I kept doing this work. And this is, for example, it's the work that is doing today my PhD student Tobias, who is now looking. So we change. Now that the machine are making it feasible, we are now looking at the sulfur, the impact of sulfur. So this is something we're doing at the moment in New Caledonia, and this is a large benthic foraminifera from here in New Caledonia, and we are trying to develop a new proxy. I mean, that's been published very briefly, but we're trying to improve that. A proxy that would allow us to reconstruct sulfate concentration in seawater using sulfur-calcium ratio or sulfur isotope of the carbonate. So that's the work of Tobias, so I'm not going to present that any further. But again, and another one, so this work, again, keep on going. And now, for example, this other student who applied with me for 2 proposals this year. It's a student, right now she's in the States, but she's willing to start a postdoc, and she applied for this proposal and is willing to work on the iodine to calcium as a new proxy for reconstructing reef oxygenation. So that's, again, a new proxy, new element. Iodine is something we need to calibrate. So she's in the proposal, she wants to do culture experiment and then field control and all of that. So, okay, so I think you understood the principles. So now I'm going to give you a summary of one selected contribution. I picked one, and because this is a very major one for me. So this is the work of one of my postdocs, so Henry Wu, who started the postdoc with me when I was still in Paris in 2017, I think he started. And Henry, he worked on a large coral core from New Caledonia. So why did we try to do that? But to start with, the first thing, of course, we were interested in the region. We wanted to make large and long pH and temperature reconstruction of this part, in particular of the ocean, but also in a very practical way, because we knew there was this massive head of coral here, c'est dans la fosse Passe d'Huitoé, and we thought, okay, and interestingly, this is not a Poraites, the one you see all over here in the lagoon. This is a Diploastrea, and the difference here is that this coral is growing only 3 to 6 millimeters a year. So meaning when you have a 6 meters big coral head, like you can see on the picture, we were thinking, oh, this is so cool. We're gonna reconstruct temperature and pH up to the Little Ice Age. Okay, we did not. But we tried, and we still managed to get a 4-meter long core. So this is exactly what you have here. Unfortunately, even if you have a 4 meters long core, it doesn't mean you're going to have 4 meters long of record, because often something happens in the life of the coral, and halfway through you can't use the carbonate anymore. But we still had the chance to be able to reconstruct continuously 320 years using this core, and this is exactly what we did. So this is how we've been able to date it, and we did some uranium thorium dating to be sure that we were really correct, because otherwise we count the bands of the growth, of the coral growth. And I'm only going to present that, and we analyzed several isotopes, and mostly, so we analyzed the boron isotopes, which I introduced before. We analyzed the oxygen isotopes. So the boron isotopes, so this is the one that you are seeing here. With the boron isotope, we could clearly see a shift starting from 1850s. So what we could see is that starting from 1850, so basically the start of the Industrial Revolution, you have this drop in pH which is continuous. So you have here your 1850 up to 2000, it's there and it keeps going. So this is also what you're seeing here, and this time we also translated that into pH value. So before 1850s, you have this data, it was going up and down, and then suddenly you hit 1850s and it goes down. At the same time, so for the boron isotope, when it goes down, it means your pH acidifies. It's very straightforward. For the oxygen isotope, what we could see this increase in temperature. I mean, this is not new. Those data have been published before with corals. The increase in temperature in coral using oxygen isotopes. This has been well published before, but that was interesting to see that measured on the same core, basically on the same samples, we had both that. knowing though that the boron isotopes and the oxygen isotopes, you don't use the same machine to analyze them. That means there is always like it reinforces the signal, meaning, okay, what you measure on this machine is perfectly in agreement what you measure on this other machine. So we had that. Starting from 1850s, we had this clear acidification, this clear raise in temperature, and the third isotope we measured, it's the carbon. And the carbon here, I'm talking about C12, C13, so the stable isotopes only. And what is interesting is that since the human has started to put back carbon into like a cycle, so through entropic activity, which is mostly using combustion, whether you're using coals, whether you're using petrol, what you do with this combustion process is that naturally your carbon, which is the lighter, meaning the C12, is going to get evaporated a lot more easily than your C13. So over the years, since the humans are doing these activities, what you have very strong enrichment in C12 and impoverishment in C13. And it's called the Seuss effect, and that again, we could see exactly that on our core. You could see this drop on C13, you could see the temperature raising, the ocean acidifying, and you could track back that this was due to carbon released from human activities. So that was a very interesting, I mean, it was a lot of work to do that, all those data and all those geochemical signature. It takes a lot of time to get there. But of course, it was the result at the end. We had a very nice publication, and it was a real success for my postdoc, who after that ended up getting a €1 million fellowship and could start his own research team in Germany, where he moved to. Anyway, I'm going to finish now on perspectives. So perspectives, so I'm going to introduce briefly two projects that we got funded this year. The first one is called the Project VAG, which is funded by the CRESICA, and it will take place here in New Caledonia. And the second project is called Pledges, which is funded by the Fonds Pacific, and this one will take place in Palau. I want to introduce these two projects at the same time because basically it's to do the same thing, just in two different areas. The objective of these studies is to investigate the evolution of microbial diversity in parallel of paleoclimatic reconstruction made on the same sample or archive. So I'm going to introduce that very briefly. So this is Palau. Another reason why we were interested in Palau, not only because it seems incredible for an environment, but also because they have naturally acidified area and some non-acidified area. And so the idea was also to go and dig some sediment there and see what we could find. But really, the idea of this project is to do this, to get this, okay, let's say on sediment core, because there's also some coral core involved, but let's talk about sediment core, to get some sediment core covering, let's say, the last 50 to 80 years, and to do on one hand with the sediment core environmental and paleoclimatic reconstruction based mostly on the foraminifera on the calcite, and at the same time from the same samples analyze the microbiome, the microbial biodiversity, to see what was the reaction of that environment quickly after an environmental or climatic anomaly. And so, of course, in the end, we want to have a model involved, and we have partners from the university here. We want to try to make a correlation between, okay, this is the climatic perturbation. What do we see as a first responder into the biology? What happens when we put it straight to it, and what is happening in the past? Had that happened in the past already, and what are the consequences? So we started on this idea, not just like that, also because I had some kind of preliminary walkthrough, a Master 2 student who did a practical with me in 2024 in New Caledonia. And what Clémence was working with was a very long core that was retrieved by the Ifremer people near Thio, literally at the exit of the Thio River. And what was interesting with this core, so we did exactly that. Instead of looking only at microbiomes, we actually look at environmental DNA, and that's what Ifremer people were doing, while at the same time I was getting all the samples and could get foraminifera all along the core. This core was covering 1,000 years. And then we wanted to match that. We wanted to match not only on one hand the species repartition of foraminifera over time, the evolution of that over time, and to correlate that with the environmental DNA data that we also had over time. And very interestingly, as you can see here, so this is, 1300, I don't know if you can see on the graph. And so here we are in New Caledonia, and here you have mining starting. So here we're beginning of probably 1930 or something like that. And it's mining starting, but it's mechanical mining. So even if it has an impact, it's not so drastic. And then comes the 1950s, and here comes the, comes the mechanical mining at that point, before it was hand mining. And with the mechanical mining starting, with environmental laws that were not in place yet, what you have here, it's this white band, probably, and I should put something like highlight the band. What you have no more forams, no more foraminifera at all. You have 10 years there, you have no more foraminifera. And if you look at environmental DNA, it's the same. You have these white things here, like this, something like over 10 years, everything is gone. And then environmental laws are put in place, and they are not allowed to leave this big, like a terril of soil after the extraction is finished, to be, when you have bigger rain that's bringing everything into the river and to the lagoon. Anyway, so afterwards, the population, they come back, and foraminifera come back, but you never go back to the original biodiversity you had before all of that. Okay, so to finish with the project we got funded this year, we are actually right now working with many other people in the Pacific. We're trying to build this Pacific hub to try to look at very different places in the Pacific. So far we have projects funded in Palau. We have some funded in New Caledonia. We have collaboration with Fiji and Australia to do kind of the same work. We have collaboration in Singapore, which is starting as well. And we have other people that we know in different places which we may contact and try to get as many samples and data as possible. Okay, so I will stop here. Thank you for your attention. And I just want to finish on something, because the 3 members of the jury that wrote a report, so thank you, already wrote a report on my manuscript. The 3 of them picked this one sentence at the end of my curriculum saying that I had a 3 career break, because I had 3 little kids. And so this is what I want to say. Thank you for picking on that sentence, because yes, it is a factor which is definitely delaying an HDR process, and this is it. Thank you for that.
Thank you.
So, thank you, Delphine, for this timely presentation that was kept just right in time.
45 minutes?
Yeah, good. And we'll start immediately with question, and I would invite Takashi to proceed on the questions, then Emmanuel, and then Cyril. Okay? Yeah.
Going back one by one.
Okay. First, can I? Okay. So thank you very much. The great talk. So I can understand how your science is growing up and spread the field from the laboratory to the field, and especially it is very impressive for me. So you utilize New Caledonia's geographical advantages, especially so at final slide you show the geographical map on the Pacific. So how can I say, the strategy of the Delphine desert science is quite important to make the future the science. So here I have 2 questions. So one is as desert indicated, there had never been an error so keen to predict the future. So what kind of the highly educated personnel are needed to, what is required for their education? So as a professor, habilitation professor, so you start to educate the PhD personnel. What is required for that? One is this. And another is the very important study you are doing, especially the social related science, the climate change or sometimes the pollution, of course the pH change. That is very important for the island people. How your result return to the society. These 2 points I'm very interested.
So, alors, the first one, I'm not sure I understood exactly the question. I think you were asking what parameters can we improve to improve reconstruction? Is that what you were saying? What is the, am I wrong?
First question is when you educate high educated student, a PhD student, what is required for their education?
The quality for their education?
Yeah.
What do I choose when I pick a student for PhD? Is that way? Okay. I think to work with this kind of work we're doing, I think you need to have people that are very patient. You need to have people that are very calm and very patient and very dedicated, because the foraminifera job can be very ungrateful. So coral a bit less, the way I use them a bit less, but foraminifera, I'm searching for students that are very curious and very patient and who likes to work with the, I guess who has this capacity to really dedicate a lot of time, to go at the very end of the task. I'm not sure it was exactly your question, but for Tobias, for example, who has just left the room, this is something I really like, for sure. I like people who are going to go at the very end of the question on samples that can be sometimes a bit ungrateful. Maybe I should put it this way. The second question, sorry, I lost your second question. What was your second question?
So what methods are being considered to return the result to society?
How to translate that to society? Well, that's a question that is being asked a lot to us at the ERD, because it's part of really our job to educate, but also to help the development and things like that. So I think, well, first of all, the first thing is through teaching. There's no question about that, through teaching, whichever age you teach, actually. And so I did not present that, but in terms of whatever, I don't like this term, but like what we call vulgarization, I do a lot of, at least once or twice a year, I try to do kind of intervention, if you want, in high school, in college. We have kids also that are coming to visit our labs at the VRD. We have the Fête de la Science at least 2 or 3 times a year to really to inform and educate, and then teaching at the university. But for me, it's not much. It's only 10 hours a year we're talking teaching at the university. It's not like I see a lecturer that have hundreds of hours. I'm definitely not there. But still, it's through teaching is one thing. And then we hear what we have, which is pretty good. We have good communication, I guess, and good capacity, because the island is pretty small. So if you can quickly involve people, like people of the Cressica. So there's someone from the Cressica here as well. It's easy to talk to them. You can meet people that have those functions, and Cressica in New Caledonia, it's all the research organism of New Caledonia. And so they can gather people on specific topics, and they can go to the government of New Caledonia. And so you have a way, you have a path to make your voice heard. I mean, on I don't want to say too much, like on a bit more than you would have compared to when I was in Paris, for example.
Okay, yeah, now it's clear your stance and the strategy. Thank you very much.
Okay, so to you it's finished? Okay. So So thank you, Delphine, to inviting me. I would like to be in Nouméa, of course, but not at 7 o'clock in the morning this morning. A little bit hard. So I appreciate your oral presentation. I was not very, how to say, smooth on my report because I had a lot of difficulty to read it, but I'm not going to speak about it. We already discussed about it by mail. I have some question, I think mainly linked to this vital effect. I'm working mainly on living forum, so it's something it's question me. And maybe the first thing is it's coral or forum. So they are linked to symbionts, and the symbionts are moving. So I'm thinking, for example, bleaching the coral bleaching, then the coral is losing the symbionts some period. Or the planktic forums, the symbionts are moving outside, inside. Do we know if, for example, during a bleaching period, if the coral is still calcifying, or if we are speaking about planktic forums, do you know if when the planktic forums has symbionts outside, if they are calcifying also, if they are calcifying all the time?
So, alors, it's in that particular case, they're very different from one another, I would say. For corals, and we have a lot more data for corals on that very specific question, because a lot of people study the bleaching of corals. And yes, I think they still calcify, not as fast, not as much, but they don't, and it's going to depend how long the symbionts are still out, because eventually they're going to recolonize the coral and it's going to, get better and start calcifying again. It has this capacity to survive a few days, probably a bit more. I don't have the latest data in mind. And I know it keeps calcifying, though. I know there is a paper on that. I'm not going to remember the first author, but there's a paper on that which shows that it reduces the calcification rate, I think. For forams, and planktic foraminifera in particular, that's a good question. The problem with planktonic foraminifera is that unless you have culture of planktonic foraminifera, which is even harder than the culture of benthic foraminifera, which you already know is a bit of a nightmare, culture of planktonic foraminifera, so far, what you have in the literature is only literally a few days. And actually, you have people in Puerto Rico. Yellow, my PhD supervisor, he did a bit of culture with planktic foraminifera. But we're talking about a week. And after a week, they were dead. And that was the end of the story. Now, planktonic foraminifera that have bleached, and whether they still calcify and whether they still survive, I honestly have no idea. I really don't know. And I don't remember reading a paper that had any idea about that. How would we know? Foraminifera, planktic foraminifera, we're talking something, they're migrating into the water column, coming to the surface to expel the gametes, basically. And then once it's done, it's going to die, lose its spikes, and sink. And if anywhere in that process, at any time it has lost its symbionts, is it still capable to do it? No idea. And I don't know how could we get this information? I don't even know.
Yeah. No, but for example, if we are speaking about corals, the aim of the question is to know if the incorporation of trace elements or isotopes is changing when the symbionts are gone, or because we can think that the actual calibration can change because there's this bleaching effect now, and this incorporation of trace element can be changing.
I'm sure it does. Again, differently for the two different organisms. So for the coral, what you're going to have an impact of your pH at the site of calcification when you expel the symbionts, because they have this regulatory effect as well. Once they are expelled, you have your calcifying center. I mean, all of that, whatever happened with the calcium. I mean, there's a lot of things that are being published proven, but still being in discussion. Anyway, my point is, I'm pretty certain you're gonna change the pH, and that makes sense, of your calcification center, or at least the activity inside, which indeed would gonna have an impact on your calcification rate, which is clearly gonna have an impact on your metal incorporation if you're talking about like, I don't know, manganese or zinc or something like that. So for corals, I would say, again, I have very little doubt about that, but again, if we're talking about a few days, with the quantity of carbonate that your coral is going to produce, your coral is producing 1.5, I'm talking about massive corals, I'm not talking about the branchia, but the massive corals, 1.5 centimeter of carbonate over a year, it's massive. So even if this happens for a few days, this will be completely blended out in the average of what you'll measure for that particular year, unless you do mensual resolution, but mostly you do yearly or monthly resolution. And if it's just a few days, even in a monthly resolution, if this has an impact, I think you won't see it that very clearly because it's just a few days. If it goes longer than a few days, your coral will probably just die, and that you will have as a black band or white band on your coral, and so you'll have a signal, and at that point that you do not pick the geochemistry, you do not analyze geochemical signal on those very bands because something has happened at that moment. So I don't know if I'm being very clear, but this is I'm sure for corals this will have an impact, and if I go to the end, I'm sure for forams as well. But again, for foraminifera, I don't know how we can see that.
It's very difficult. No, but it's quite an important question when you saw on the last presentation the result of your postdoc with the decrease of the pH and how to. It's sure that the pH is decreasing in the seawater. This is not a problem, but how to differentiate this kind of vital effect.
No, that I'm sure actually, because I've asked myself this question a lot, and you can really see, you actually on a coral, you can even, when you have a lot of money to do geochemical analysis, you can even do a diurnal cycle, because the pH within your reef, your pH is going to fluctuate a lot on a daily basis. And when you can, because they produce so much carbonate, you can actually make very small, very high resolution, of analysis on your boron isotope. It costs a lot of money, but you can do that. And you'll see this bending, you see your diurnal cycle. But at the end, what we're showing here on the paper with Henry, like over 300 years, you see those fluctuations, you always see them. You have them monthly or literally seasonally and all of that, but this is not what we're looking into. We're looking about the trend, and the trend over 300 years is literally undisputable. There's no question about that. I mean, and the diurnal, that's where I was going to go. The diurnal fluctuation we see, this is exactly that. That's the symbionts activity during the day and no activity during the night. And that's the fluctuation we already see.
Yeah. And you work a little bit also on the incorporation of metal due to pollution. And do you think that it can be also influence some incorporation of trace element in the shell? So imagine that you have a high pollution of one metal, it doesn't matter which one. Do you think during the calcification it can also change the ratio between the elements you want to measure? Do you understand my question? Not sure. Yes.
I think so. It's because most of metals we're gonna find in seawater, they have this 2+ form, and they're gonna take eventually the calcium spot in the crystallization process. But what I noticed though, because we worked, it was very interesting during my PhD. I had this, so the other papers that I showed that were published during my PhD, where I'm not first author. There was actually a colleague, and he was using my culturing facility because built that and I put that in place, and he asked whether he could use it to look at the concentration of heavy metals as you would find on at the level of the Medio-Oceanic ridges, so like pretty heavy metals, like in pretty heavy concentration in many, many metals. So he used my culture experiment to do exactly that. And what we could see most of the time for heavy metals, yes, they're going to play a role, but what they do mostly is that they very passively incorporate it until they kill the cell. Because the forams, this one cell, it was Ammonia tepida again. So they are incorporated in the exact, basically, proportion. There's always a bit of an offset, but not huge. The proportion, the concentration into the seawater, I'm talking about copper, I'm talking about barium, I'm talking about, like many of them. And they like very heavily dependent of the concentration you have into the seawater until they become lethal for the cell, and then everything dies. And that I'm talking about mercury, like there were things like that. And before that, they just like, if there's a lot in the seawater, there's a lot in the calcite. There's not much in the seawater, there's not much in the calcite. Surprisingly, there was kind of the main effect there was.
Okay. And maybe I'm going to ask a question on your project, and if you can put the last figures with the forams and the microbiome, and you can see this level without any forams and any microbiome. So now I understand a little bit better your project. So your project is to do the diversity of microbiome and to calibrate them in the time using foram proxy. But do you want to try also to? It was what I understood when I read the project. I understood that you want to make a relation between the microbiome and the forams diversity, or is it really separated?
It is yes and no. I mean, it will depend how it goes. I want to say, to start with, no, it was in the other sense. I mean, we want to first reconstruct the thermic and then the environmental anomaly using because what I do not show here, I only show, yes, it was foram diversity, species diversity, that we plotted against environmental DNA. But what we had though as well, I had the magnesium-calcium ratio analyzed on the little shells. I have also I had the full chemistry of the shell. But we had a problem with our ICP-MS, and some of the data I don't trust 100%. So I don't want to show that. I want those forams to be reanalyzed before they can be plotted versus the environmental DNA. So far, the one data, we were 100% sure that species repartition and evolution over time. The other two projects, the idea is first to reconstruct thermic anomaly, like so temperature, but also heavy metals. And once we have those anomalies that are, because in different places they're going to be at different moments, and once we have literally really located and recognized this anomaly, then look at what was the impact of this anomaly on the microbiome. It was more in the other direction. And same with corals. That's why we are going to do also coral cores, because the coral, they should allow us to have, okay, it's not the exact same archive, but they're going to be taken in the same environment. And we have because corals, this one and only organism, we had a more continuous reconstruction of environmental parameters. So it's more in the other sense. But depending on what we find in the cores, and if we have rich diversity in foraminifera, that can also go in this sense as well. But right now, I don't know yet.
And do you think to also analyze, maybe it's the last question, the assemblage, the diversity of forams? So here you have absence occurrence, but do you think, I know it's a lot of work, but do you have the project to look also on the community of foraminifera?
So only species repartition? This is what you mean?
Yes.
In the coral cores?
Yes.
In the sedimentary cores? So it's not described like that in the project, but again, I think it's the starting point of these studies. Like, yes, at the beginning, I'm going to search this one species that's going to be all over the core because I want to follow one parameter, and so I need one all over the core, and often it's ammonia, or it's it's always the same 3 species. Like, and Marginopora is not so bad depending on where we are. But so that's the first thing. But what we did with Clémenceau, which he ended up doing, and there was the question of Takashi, because she took that on herself to go for precise, like species determination, and thanks to also Tobias, who really helped, because after doing work with you in Angers, he was really good at species determination. So they really worked, the two of them, together to determine all of these species along the core. And the data we have today, it's thanks to them, because there would be otherwise only geochemical signature. And as I said, I don't really trust them. So right now, I would not publish that. And the paper is actually it's under review for Nature Communication. It's going to be a good paper. I think it should get out in the next few months. Yeah.
Good. I have still time or no?
Yes. Yes.
Yes. The last one I have to choose in my. Yes, maybe this one. Still scientific. I have other question after, but you are speaking about ammonia. And you know that with phylogeny and the DNA improvement, then we have a lot of phallotype. You know the story with T1, T2, Tobias no, T6, and they are looking very similar, but they are not the same. Do you think how it can influence your record? Is it important to use really the same phallotype?
If I'm being really honest, I don't think it does. I don't think it has much of an impact. I think we are talking about species. Today I was actually thinking about that when I prepared this publication, because most of my PhD papers, I did it, you clone the forams. You've got reproduction, you clone them and things like that. So I did not, because we're not very successful with reproduction, but they're still all coming from the same environment, same type of forams. Someone that took over the culture experiment that I built at AVI, he actually cloned the forams, and he got this DNA literal, like, how you call that, like a real proper sequencing of those exact forams that he had been cloning and reusing for the experiment, and he renamed them. So there wasn't ammonia tepida, it was ammonia, I can't remember the exact number and things like that. I honestly, I think if you are on a species which is different enough, in a sense that it's just it's not in terms of looking, but it's definitely how they live and how they build their carbonate, and even between an ammonia beccari, for example, and an ammonia tepida, and I often had both in my sediment, and I looked at that already on the geochemical signature, and it has an impact, but I think it's because beccari is much bigger. Like the room, the chamber, for example, produced was much larger. And what I think has an impact here, it's the calcification rate. And it wasn't a drastic impact. It was just a slight, if you were really looking very carefully, you could see, okay. And I think what we're talking about here is calcification rate. But that's always going to be, in between two species, there's always going to be something like that. But in between two ammonia, tepida, and I know, like the different letters that they've named them and things like that, I really don't think so. And I have kind of for proof for that is that I used the equation I established on ammonia tepida from northern Germany, so it was Dorum. And I used the same equation to reconstruct temperature on different ammonia tepida from the Atlantic Ocean and things like that. And it worked. It worked very well.
Okay.
The one it didn't work, it's here in New Caledonia. It doesn't work at all. And I did it with ammonia tepida here in New Caledonia. Absolutely not working. So there's something I need to figure out here. I think
maybe they have not the same physiology, or there's something.
It's very weird, and it's actually very interesting. I need to reanalyze a lot of ammonia from this same core, exactly like the CHO core, which I introduced, and the equation doesn't fit at all. So I don't know.
Okay.
We have to look into that.
And just to end, so it's funny because Toyo, he asks a question I want to ask about the quality of the PhD, but then to close the discussion with the Visio person, I'm going to ask you the question that what do you think is the most important quality when you are supervisor?
Oh, it's curiosity and determination. My quality or their quality? Mine?
No, the quality of the supervisor. So, meaning you and me. What's the opposite question of to you?
Okay, so it's patience. Yes, it's patience.
Patience.
Okay. I would say patience and let me think. Yeah, I think it's respect to start with, but that is not just science; it's life. You know, like you need to respect people.
Okay.
And then it's patience in the sense you watch them grow and they get there, and sometimes you want to push them there, but you can't. And but yeah, I think patience and explaining again. And to go back and explain again when they don't understand. I think this is the key. I'm not sure I had that with my PhD supervisor, who you know pretty well. Patience was not his, main quality, but he had other. And I think long-term vision, I have a global and long-term vision. This is something you can't expect from them during a PhD. It's too early, it's too soon, so you need to think further. You need to help them build that as well. I think this is quite important.
Okay. Thank you very much. I think I did more than 20 minutes, but thanks.
Thank you, Emmanuel and Cyril, if you want.
We lost the public.
Merci beaucoup, Monsieur le Président. Merci, Christophe. Chère Delphine, thank you very much for having me in this jury. I really appreciate your work since your PhD, and I specifically like that I found that you have a backbone in your research. You always deepen the scientific questions, trying to find new proxies, calibrating them. You did not diversify too much your research, and it's something that we can feel focused and with lots of ambition to develop this stuff. So thank you very much. I also like when a scientist is able to do a step back and to analyze what he has done, his way of thinking, his way of doing sciences. And in your manuscript, you wrote, on a more personal point of view, this work has also deepened my own scientific reasoning. Experimental manipulation of living organisms forces us to confront the limits of our categories. Yeah. Laboratory conditions are always simplification of natural environments, and yet they reveal processes that are otherwise invisible. Yeah. Can you give us some example and detail a little bit this sentence, please?
Oh, this is so true for a sentence. I can give you examples, so many, but it's a snapshot. What you do in culture experiments is just a snapshot of what your imagination really is building in terms of what can happen into the environment. The limitation are like millions, and the limitation, I would say, it's all the things we cannot see that are happening in an ecosystem, which we obviously cannot put in culture, because we're gonna look into this one organism. In my case, so that was coral or forams, but forams in particular, the one single one-cell organism to live and survive, and the corals as well, but there's so many unknown interaction happening right there, virus, bacteria, and maybe this is why I'm going there now, actually, and this is the key and the path, because there's so many things happening, and people working with corals, you have people specialize in so many different specificity, and they all work, like we all do, like very specialized in this one thing, and they struggle to work together and bring all of that together. And that's exactly, for me, the limitation of culture experiments, is that you, of course, what you're looking at, you're looking 100%, but what is everything you're not looking at, and what is you're not seeing? So limitations, so yes, I can give you examples of limitation, so many, like the number of times with corals that we did not, for example, consider exactly that, the symbionts impact. When we're just looking at boron, we could not actually, there are parameters, we could not look at everything and say, okay, we need to actually almost make them bleach at some point to be sure that we just want to see what is cascading, which is a complete scientific anomaly and stupidity, because this is massive stress for the organism, which is not what you want, because it's going to start to calcify like crazy, and which is going to be wrong for your geochemical signature. But sometimes to answer a question, you need to stress them so much that they get to this breaking point, basically. So that's the limitation for me of your culture experiments. You cannot reproduce what's happening in the nature. There's no way.
Okay, thank you very much. To perform your work, to perform your research, you must have a wonderful natural environment, what we have here in New Caledonia,
Oui.
with beautiful lagoon and lots of different environments. And you also must have mesocosm facilities and also state-of-the-art facilities. So you showed us some examples at $2 million, you say, analyzer. So do you consider that it's better for a scientist working on your topic to be close to the field, close to mesocosm facilities, or close to the state-of-the-art facilities?
It's a very good question. I think it depends. I think it changes sometimes. But for me, now, at this point of my career, it's better to be close to the field. Because now I built a network which is, let's say, vast enough and strong enough, and I know the people, I worked with them in their lab, I saw them with their machine, how they worked, and I trust them, not all of them, but I know the people I can trust, I know the people, exactly that. And so based on that, and again, it's 15 years or more since I started my PhD, it's 2005, to be able to build that. It took me a long time, and now that I have that, the limitation, the analytical limitation I have with my own lab, although we're pretty lucky, really, we're getting a new ICP-MS in 3 months and things like that, but yeah, for sure, we're not going to get a multi-collector, we're not going to get this literally like, the latest state-of-the-art machine, but at the same time, we have these incredible samples, and it allows me also sometimes to negotiate with people all over the planet and say, listen, I'm sending you these samples. We can share a student. I know you can do this and this and that. And so now for me in my career, it's better and easier to be close to the field. But it was not always the case.
Okay, thank you. So we will not buy a multi-collector, but...
Well, you can.
No,
I mean...
I can't. But do you think we can have new specific mesocosm facilities in New Caledonia? I mean something bigger than what we have.
So, well, yeah, and I think, so it really, again, depends what you call mesocosm facility. I know at the ERD this is in discussion a lot, that they want to build a culture facility. So mesocosm, for me, it can be, for example, the LEMA, they have mesocosm facility, and we've used them, so the IFREMER, and we've used them, but it's limited because it's not made for our organism. It's not made for foraminifera. It's made for algae, microalgae. So we use them, and we've been quite annoying because we want longer experiments. We want to use them differently. So yes, I think we should in Caledonia. I think that would be used. There's no question. There is a need for that. The question is where, maybe, and who would be running that. I think mesocosm facility everywhere, it's always a win if you have the right people to run the facility, because it's a 24/7 job. You need to have someone there, a technician, an engineer, all the time to run to survey that. And so I know that, again, that's been discussed at some point at the university. I know it's been discussed now. We're pushing for that at the ERD. I'm not sure this is ever going to happen, but yeah, that would be incredible for sure. I mean, I'd love to use that.
Thank you very much. And now I'll ask you some questions about mangroves and foraminifera in mangrove. So I didn't know, it's not my specialty, so I didn't know about delta 11 boron and the ratio boron and calcium. And in mangrove, there's different... I have two questions. One concerning diffusion of CO2 from the pore water from the sediment to the water column.
Yeah.
And my second question is, sometimes the canopy is open and sometimes the canopy is closed. So there's different light intensity reaching the surface of the sediments. So do you think that these two parameters, so light intensity and CO2 diffusion, may be recorded by benthic foraminifera and the proxies you're using?
No, I don't think so. I think not this way anyway, because you're on your daily cycle, basically, again. And foraminifera, even the large benthic foraminifera, which produces a lot of carbonate, no one can tell what's going to trigger the production of this carbonate at this exact moment. And I've been observing them so much for so many years. And at some point, they're not happy, and suddenly, poof, they produce a lot. And we don't really know why. It can be food, it can be light, it can be temperature, it can be so many things. And depending on the species of forams, they are going to produce 1, 2, 3, 4 chambers just at once like that. My point is, the strength of them is it's the average, is averaging the signal of this environment, not using them as you would like for a couple of hours or even a daily signal. I don't think we're there yet because we do not understand yet. I'm not sure we will ever understand exactly how they produce that. It's not like I would say corals are a lot more regular. Forams, from what I can see, it's very irregular and it's uncontrollable. It's just, we don't know. And based on that, I don't think we can monitor that on a daily basis. But what I think we can do, we can compare two environments. Let's say you have one environment of mangroves where you have, I'm not going to use the right term, canopy opening and closing, but let's say more often, and with more, either one way or the other, more carbon arriving to the sediment or being diffused to the atmosphere. And you have another mangrove, or it's the contrary. What you can use your foram for is to compare these two environments, because again, they're going to give you an average value, not something over a few hours or a few days, but a few months. And they're going to tell you that. They're going to say, okay, this one in this mangrove, it got more of this and more of that. This one, it got less. And that's, I think, how we should use them and how it's being used, because it's trustable. In the other direction, I would think it's complicated.
Okay, thank you. I have a second question about mangrove. So I talk about mangrove sediments now in tidal creeks within mangrove. So because of the tidal pumping effect, in tidal creek, the water quality is very specific, with sometimes several thousand ppm of PCO2, with really high salinity, very low oxygen concentration. So it's kind of multiple pressure on foraminifera. And do you think it can have an influence on their development, their diversity?
Again, depends on the species, but how much water are you talking about? Are they going to be immersed at low tides?
All the time, yeah. In tidal creeks with water all the time.
Water all the time.
Even at low tide.
So it depends on the species, but what I know from those foraminifera is they're extremely resistant. They're so resistant, like it's almost a pest sometimes. They're like everywhere. Like if you find one species left, it's going to be one foraminifera. And so, yes, this is going to impact them. It's always a threshold thing. It's going to impact them. So the 3 parameters you just cited are interconnected, and that are what we're observing on a global scale, I guess in proportion, that are a lot more important in tidal creek. I don't know. Again, until you hit a threshold, otherwise they'll survive there. That's going to impact their calcification, that's going to impact their survival rate. They're going to slow down. That's what they do. They slow down. They have also often the capacity to put themselves a bit like, in dormance, like benthic ones. The deep benthic ones in particular, sometimes you feel they're dead, but they actually have been in dormance for I feel like a couple of years even. So they have this kind of very strong resistance. But yes, to answer your question, whether this is going to impact them, that will impact them for sure. Whether this is going to kill them all, I'm not sure. Well, clearly, they managed to kill them all for 5 years with whatever. This, more I think about this graph, I think there was one heavy metals again, that hit the threshold. This is what I think. Which heavy metals, and maybe two of them with nickel and chromium or barium, cobalt, I'm not sure. But I think this is what we're seeing here, that hit the threshold, and that's why they disappeared. More than everything else, like more than a stress of calcification, everything is just, this is it.
Okay, thank you, Delphine. My last questions. You said that for the moment, the best place for you to be is New Caledonia, but in 10 years, where do you think we'll be, and on what kind of project you'll work?
I have no idea. I'm not gonna lie, I have no idea. And this is what I like about my job, is that I have no idea. And I think I really enjoy this job, like this whole job I'm doing with proxy development and calibration validation. I really enjoy that. I'm a bit frustrated because I haven't been able to do a lot of C campaign lately, and it was more because of personal reason. And so I want to change that. I think I want to write a program, and I want to know more about the Plantic foraminifera also that we can find around New Caledonia, because there's nothing in the literature. So I could imagine to have a bit of a switch like that, and I really want to keep going with this work that we've started and implemented with this Pacific Hub, because I find fascinating to look at all those very specific endemic environment of the Pacifics, which are threatened, obviously, but also incredible, the richness and the biodiversity of that, and this is fascinating. So yeah, I think that's the two ways I see in terms of scientific questioning. I'm sure there'll be something more until then. Where I really see myself, that I really cannot answer. I really don't know.
Thank you very much, Delphine, and again, congratulations for the chapter.
Thank you.
Thank you, Cyril. Nazar, it's your turn.
Merci, Monsieur le Président. Désolée, je vais parler en français. Donc, je te remercie, Delphine, de m'avoir invité à ce jury, même si je ne suis pas vraiment dans le domaine, mais ça m'a vraiment très intéressant. Je trouvais que c'était très intéressant. J'ai pu comprendre quelques trucs, pas tout, mais l'essentiel en tout cas pour les points importants pour les futurs travaux auxquels je serai associée avec toi. J'ai trouvé tes démarches scientifiques très intéressantes, avec une continuité vraiment thématique. Tu as une maîtrise exceptionnelle des aspects méthodologiques, analytiques, comme le proxy, les incertitudes, calibration, etc. Un positionnement stratégique sur le Pacifique qui est très important, avec un impact scientifique et social. D'ailleurs, j'aurais une question sur l'impact social. Une interdisciplinarité qui me semble importante dans la recherche, avec une dimension internationale efficace. L'articulation entre recherche fondamentale, instrumentation et application climatique est particulièrement convaincante. Au vu du parcours, des publications, des projets, de la continuité sur le sujet, des encadrements, le fond me paraît extrêmement solide pour l'obtention d'une HDR. Vous avez vraiment des approches intégratives sur une longue échelle de temps combinant différents types de données, donc une hétérogénéité des données, ça c'est très important, et ça nous intéresse beaucoup en data science. En tout cas, disons que les jeux de données sont très intéressants. Je suis patiente de les voir.
Moi aussi.
J'ai une ou deux questions. Je voulais savoir sur l'impact social. C'est quoi en fait? Est-ce qu'il y a des collaborations avec des chercheurs en sciences sociales
En sciences humaines et sociales? Pas plus que ça. J'ai pas plus que ça de collaboration en sciences humaines et sociales. C'est quelque chose, je pense qu'avec les projets qu'on a eu de financer ici et en Calédonie et dans des zones particulières du Pacifique, ça va en découler. Je suis pas inquiète par rapport à ça. C'est pas quelque chose qui a été construit. Enfin, nous, on en a beaucoup discuté parce qu'on a soumis aussi d'autres projets, notamment à Nauru et aussi en Australie et à Fidji. Et effectivement, il y a des endroits où ça se met plus facilement en place que d'autres. Maintenant, oui, évidemment que ça fait partie de l'équation. Évidemment qu'aujourd'hui, on ne peut pas faire de la science que pour faire de la science. Et en tout cas, nous, à l'IRD, on est fortement encouragés à le faire comme ça, mais pas le faire comme ça, justement. Et je pense qu'aujourd'hui, on est... Non, donc j'ai pas d'implication avec des chercheurs en sciences sociales. Ça me fait pas plus peur que ça non plus, dans le sens où je pense que ça peut se mettre en place quand on aura les bonnes données dans les bons endroits, avec les bonnes zones. Et quand on en parlait, par exemple, avec le projet Cressica, donc avec les zones à UVA, où on peut aller sur place et discuter avec les gens. Bon, de façon générale, je pense qu'avec toute la transmission et la communication qu'on fait déjà, la science sociale, elle est là, même si, attention, on ne fait pas de recherche en sciences sociales.
Ça peut être une sensibilisation.
La sensibilisation, elle fait définitivement partie de mon travail. Ça, je ne dis pas ça vraiment... je vois Florent là, donc je pense aux interventions de dernière minute où il m'envoie discuter, faire la librairie humaine au centre Chibaou, par exemple, ou des choses comme ça. Mais voilà, oui, ça fait partie, c'est à travers ces actions-là, plus qu'à travers des chercheurs en sciences humaines et sociales. Mais encore une fois, avec plaisir, quoi. Je pense que c'est quelque chose qui peut se mettre en place.
Merci. J'ai une deuxième question. Vous avez parlé de la correction des incertitudes qui se propagent et qui peuvent être... Avec cette correction que vous avez établie, est-ce que vous pensez que cette limitation est suffisamment atténuée ou peut-être qu'il faut encore regarder cet aspect d'incertitude ou pas?
Alors, je pense que on a fait ce qu'on pouvait faire avec les données qu'on avait. Donc, je pense que là, on est arrivé au bout. Alors, c'est un papier. Là, je ne l'ai même pas présenté parce que, à la fin, j'avais une présentation qui était beaucoup plus longue et j'en ai enlevé beaucoup parce que ça ne tenait pas dans 45 minutes. Mais effectivement, j'ai même des diapos là-dessus. C'est les error propagation du papier, où on essaye d'appliquer les proxys qu'on a calibrés en amont avec des foraminifères planctoniques. Et effectivement, dans ce papier, on a vraiment joué avec les propagations d'erreurs puisqu'on a essayer d'imiter ce que font les gens aujourd'hui en paléoclimatologie à partir de foraminifères qui viennent de carottes sédimentaires pour reconstruire une température passée. Et là, l'idée, c'était de dire, nous, on a même des forams parfaits, c'est-à-dire qu'ils n'ont pas du tout été soumis à la diagénèse, ils sont vraiment parfaits. On établit l'équation à partir de ces forams-là. On a des équations qui ne peuvent pas être plus adaptées à nos échantillons, et on va jouer ce jeu d'imbrication, de reconstruction l'une dans l'autre, en utilisant le magnésium-calcium pour reconstruire la température, cette température reconstruite qu'on injecte dans les températures de O18 pour sortir l'O18 de l'eau de mer, ce O18 de l'eau de mer qu'on va finalement utiliser pour reconstruire la salinité. Donc voilà, ça c'est ce qui est fait aujourd'hui dans la littérature. Nous, ce qu'on voulait prouver, c'est qu'en ayant les échantillons parfaits, les équations parfaites, à la fin, les données de précision de salinité qu'on a pu reconstruire, c'était plus ou moins 5 pour 1000. Pour quelqu'un qui fait de l'océanographie, plus ou moins 5 pour 1000, c'est à peu près toute la salinité de l'océan. Voilà, ça va aller de vos zones... Bon, dans les zones côtières, elles sont vraiment... peuvent être beaucoup plus salines, mais voilà, c'est 5 pour 1000, c'est pas une reconstruction crédible. C'est pas quelque chose, on peut pas... C'est beaucoup trop large en termes d'incertitude. Donc voilà, ce qu'on a voulu faire, c'est ça. Par contre, ce qu'on a démontré quand même en faisant ça, c'est que pour les deux autres paramètres d'avant, donc le paramètre final salinité, et je pense que le message du papier, il était vraiment là, arrêtez de faire ces reconstructions de salinité, n'allez pas jusque-là, arrêtez-vous au delta O18 de l'eau de mer, qui est déjà un facteur ultra intéressant quand on fait de la paléoclim, puisque là, finalement, notre incertitude, elle était quand même raisonnable et acceptable. Et en tout cas, ça donnait une information intéressante en termes de paléoclimatologie. Et par contre, la température sur le magnésium-calcium seul, donc un proxy direct, là, on avait de très très bons résultats, quoi. Donc finalement, est-ce qu'on peut aller plus loin avec ces données? Non, je pense que là, d'ailleurs, moi j'avais demandé à des collègues qui étaient... qui avaient d'autres spécialités, parce que moi, j'avais pas les connaissances en mathématiques suffisamment poussées pour aller avec ça, mais vraiment, je pense qu'on a fait ce qu'on voulait démontrer. Voilà. Ça ne veut pas dire que c'est le cas à chaque fois, et surtout, ça ne veut pas dire que ça ne peut pas être fait dans le futur, mais il faut des bases de données un peu plus importantes. La limitation, souvent, là, ce n'était pas l'expérience de culture, mais souvent dans les expériences de culture, c'est qu'on n'a pas un gros jeu de données. Et donc, à partir de là, aller faire... même de la propagation d'erreurs, etc., c'est vite, ça s'arrête vite. Et d'où l'intérêt, finalement, avec les carottes sédimentaires, là, on peut sortir beaucoup plus de données. Et je pense qu'il y a des choses très intéressantes à faire. Voilà.
Ouais, j'avais une troisième question, mais je pense que ça a été un petit peu posé dans le... Peut-être par rapport à l'étude dans le Pacifique. Pensez qu'on peut généraliser ce qui se ferait dans le Pacifique ou pas du tout? Est-ce que le Pacifique, c'est vraiment particulier
Le Pacific Hub, la question pour les comparaisons d'environnement
Ouais.
dans le Pacifique. Non, je ne crois pas qu'on puisse généraliser ce qui se passe dans le Pacifique. En comparaison à d'autres environnements, d'autres océans, c'est ça? Ou je sais pas.
Oui, par
Je ne
exemple.
pense pas qu'on va trouver quelque chose qui sera comparable pour chaque île, entre les îles du Pacifique. Je pense qu'on va trouver des choses très très spécifiques à chacun de
À
ces
chaque
environnements. Ouais.
D'accord.
C'est ce que je pense. Maintenant, il y aura des similitudes aussi, forcément, mais je pense qu'au contraire, et c'est ce qui m'intéresse, moi, c'est l'endémisme de ces environnements, c'est de voir jusqu'à quel point, c'est parce que, mine de rien, même si effectivement, ils sont tous dans le Pacifique, mais bon, on parle quand même de centaines, de milliers de kilomètres pour des environnements insulaires qui sont tous baignés par des courants différents, qui sont tous soumis à des paramètres différents, qui ont tous des potentiels... que ce soit des perturbations environnementales ou climatiques différentes, et qui sont ultra spécifiques à leur propre environnement, justement parce qu'il n'y a pas de communication ou peu de communication avec le reste des environnements de la Terre, quoi. Donc je pense qu'au contraire, on va trouver des trucs très très spécifiques en termes de biodiversité. En tout cas, on le sait déjà, on a des organismes très spécifiques dans ces environnements. Maintenant, des réponses à des perturbations globales, ça je sais aussi qu'on les trouvera. C'est-à-dire que typiquement ce que j'ai montré là sur le pH, sur exactement ça, c'est-à-dire l'augmentation de température, la baisse de pH, et être capable de retracer ça avec le Suess effect, avec des processus de remise en circulation du carbone par des activités anthropiques, ça on le retrouve dans tous les océans de la Terre, quoi. Que votre carotte de corail, elle arrive du Pacifique. Moi, j'en ai qui viennent aussi de l'océan Indien. J'en ai qui viennent des Caraïbes, etc. Même si c'est pas forcément exactement la même espèce, on retrouve la même chose partout. Alors, avec des tendances qui peuvent varier, fluctuer, être plus ou moins fortes, etc. Mais les tendances, elles sont globales, hein. Donc ça, oui, ça, on va le retrouver aussi dans les zones du Pacifique.
D'accord. Bon, ben voilà, merci. Je voulais juste faire un petit commentaire disant que je suis contente d'être là et ça m'a permis un petit peu de voir les éléments sur lesquels on va peut-être être associé dans les futurs travaux. Et je suis impatiente, comme je l'ai dit tout à l'heure, de découvrir ces jeux de données qui sont originales et qui sont pour moi complexes parce que elles sont hétérogènes et c'est un défi aussi pour trouver des modèles.
Ouais, je pense qu'il va y avoir un peu de travail. Je pense que ça va être un défi. Je pense, mais ça peut être très intéressant. Ouais.
Voilà. Merci beaucoup.
Merci.
Thank you, Nasa. So I have the honor to finish this. Thank you, Delphine, for your presentation, and thank you for inviting me. It's not my domain, as you know. But as a climatologist, I've been hearing about proxies and paleoreconstruction since my PhD. I was in the same lab as you initially, so I got the chance to talk with some of your colleagues. So for me, I have a few questions. You mentioned, and we always mention to students that we look at the past to navigate the future.
Yeah.
This is your sentence.
Yeah.
And I'd like you to explain to me how this past that you've been looking at navigates the future. For example,
Yeah, navigates. But yeah, no, it is actually, because I was going to say it's not the right word, but it's. For me, it's what I introduced with the three different examples of climatic anomaly in the past. And for me, how it navigates the future, but for foraminifera, for example, okay, can I, up. It's gonna go back here. Sorry, it's a bit, I cannot get out, otherwise that cuts the connection. So I'm getting there. I'm going back to this climatic anomaly. So navigate, it's informed the future more than navigate. So navigate, if we well, which we eventually we will, and we definitely should put in place laws that change the future. But what I mean with navigate in that case is more how do we, the information we get from the past, how can that help us to understand what's happening at least right now? Future, it's again another story because I don't have a crystal ball or something. But yes, what I wanted to show here was this graph for us understanding here. So like in the sediment core, you can really see this anomaly, and this is very interesting. You have some benthic anomaly and you have some planktonic anomaly, and you see how the perturbation has happened. It's going to have an impact either on the planktonic community, so in the water, or in the benthic one. And this is something, unless you look at that in the past, you cannot understand when it's happening to you right now. And was really interesting to look at that. So because I had colleague, I only looked into that because I have this colleague in Paris who is a lecturer at Sorbonne University, and we shared a PhD student, and she specialized in foraminifera, but in paleoforaminifera, so nothing with the modern one. And I learned so much from her because of all of that. When reading a core, like a sediment core, we get all the IODP, cores and things like that. And for her, she can read the core so clearly, and that's the term. She can say, okay, we are in this anomaly. We know here this is, okay, I'm giving like stupid example, but that's a temperature. That's what happens when temperature hits again. Often it's a threshold. It's always matters of threshold. So you hit a threshold. Here we know it's like methane. That's been, for the PTM, it's methane that was going to be degazed massively, from the carbohydrate of the ocean. I mean, not carbohydrate, carbon anhydrous of the ocean that was starting degazing methane a lot. And all of that had an impact on the carbon cycle, similar to what we're having now, but still a bit different. But anyway, and so that, you could really read that into a sedimentary record. And how does it help to navigate the future? Well, it gives you big hints. You say, okay, this is what happened with the carbon at that point. The next thing was that, and look what happened into the biodiversity. And in that case, for example, so the PETM in particular, that was the benthic community, and those are the organisms that disappeared first, this and this and this and that, into that thing. So now, does it give me key to solve what's happening? No. Not like that, but it gives you big hints and understanding. Yes, that's for sure.
Okay. So for the future,
Oui.
as you know, we use models.
Oui.
So what's your models like
Yeah.
PEMIP comparisons, CMIP comparisons, climate models, Earth system models? How do you place your work in this modeling environment? I mean,
how do I what?
How do you set your work? How would you interact with that? Because you do not seem to interact much
with modeler.
Whereas that could be used.
But that's actually a very good question. What I noticed, though, is that my data, once they're being published, then they are used by modelers. But when I tried, because, again, we were in the same lab, and, there's a lot of modelers in this lab, and when I tried to ask them clearly what would be interesting for them and how, in any way, because they knew what I was doing for a job, what could I do and things like that, I never got a clear answer otherwise, or something like, I don't need data. And I was just... Okay, well, if you don't need data for your models, this is interesting. So I understand, though, their point of view. I see as in a much larger scale and much more global, and sometimes very small data on a very specific place and environment and time and things like that might not be that interesting. I noticed, though, no, I don't have. I tried. I really tried, but it never really. It never really got better than that. And apart from statistics and what we're trying to, but that's different. Like this is us building a project together. And we, for example, with NASA, we went to solicitate her and say, okay, listen, we have a problem. We don't know, we want to be better. in terms of correlation for this and this data. Can you help us with that? Which is also what I did with you when we worked together at some point. I had a problem and I went to see you, but it doesn't work in the other direction. It means I cannot go, I tried. I went to see them and I say, what can I bring? Because they don't come to me, which again is fair enough, but I tried to go to them and say, what can I bring? And I didn't seem to be able to bring anything interesting.
Okay. It's too bad. But I've seen...
the door is open. I'm happy to.
I've seen, Emmanuel, your hand raising. I have a last question. Maybe you want to say something after my last question. I'll give you the floor again. So my last question... sorry, I'm just finishing. My last question is, okay, in these new projects, you mentioned this, and this is, I know it's very fashionable, but I'd like to know your answer on what kind of IA model or machine learning model are you, because this is mentioned in your future work.
This is NASA.
But I'd like to have your answer on this. You are planning to do some modeling, machine learning, IA modeling. And my question was, when I was reading this, but what are these models based on what? And what in situ data, environmental in situ data, are you going to use? Not only the one you're going to measure, but the in situ.
Two different things. So in situ environmental data are data that we plan to do in some. So the two different projects are not exactly the same. In the first one, we plan to do a bit of a survey for a few weeks and a few months, where we're going to measure all in situ data and take samples both of microbiome at the same time, and me also on some pieces of corals and foraminifera to make calibrations. So that's the in situ data I was talking about. How can machine learning and artificial intelligence help us with what we want to do? Is definitely in the size of the database we're gonna get. And we're talking in microbiology, and that's not, again, my specialty, but with Veronique, when you do sequencing, and this is what we're gonna do, we do sequencing over sediment cores, and not only in modern environment, but also in the past, the quantity of data you're gonna get out of this sequencing, we need help with. And that's where we went to knock at the door of NASA and say, we're gonna need someone not only to do something with all this data, because it's a real full-on job, but also to correlate that with other reconstruction, which we'll do in parallel. How exactly? I think this is me in my head, and yes, I'm not a modeler and I'm not specialized in artificial intelligence, but in my head, this is where I see that as something, as a facilitator. I say, okay, we have this massive set of data. We need something to facilitate the correlation.
Okay, we'll leave that to NASA and future results. I was more thinking about in situ hydrodynamical data.
But no, we don't have hydrodynamic data. We don't have in situ. I mean, we will have what we measure, but we don't have. Otherwise, you have what we have in the.
Because you will need to calibrate. You will need to calibrate your proxies anyway, so you will need in situ data. No?
I have pretty good calibration, or for corals in particular, and for the forums with the marginal power, what we're doing with Tobias. Calibration, I already have them. So no, I don't need to calibrate them more than that. It depends which parameter I want to reconstruct. But no, a few weeks or a few months of calibration, that should be enough if I need an extra calibration for something. But right now, I don't even think I need. I mean, it was more for the microbiome part, the calibration.
Okay. So, Emmanuel, do you want to ask a final question, maybe?
No, I just want to add a small thing about the forams and the mangroves. So the mangroves are so much acidic and so much refractory organic matter. They are mainly agglutinated forams, which are not very useful for calibration.
No, we have some, and they are not agglutinated, and that's what we've been doing with Tobias. We have Marginopora, not as many, but we do have a lot. It depends where you are in the mangrove. This is really true.
Yeah. No, it's true.
But the interest also with the mangrove, I mean, for Tobias looking at sulfur, the interest with the mangrove was the H2S, which, can be released from the mangrove, was to try to see whether we could find foram getting closer to that. To be fair, it's been difficult. Yes, for sure. Like we don't have many species. We have only a few that are surviving, but we do have some that are definitely not agglutinated.
Okay,
Yeah.
good. And I would like to add something also that I think it's important. Thank you, Delphine, for your enthusiasm also. It's very nice. Thank you.